Okay, let's kick things off. Imagine this. Fixing damaged tissues inside your body, but without any surgery. Just printing them in place.
Yeah, it sounds like science fiction almost. But there was this really interesting piece on Singularity Hub back on May 12th, 2025.
Right. About something called DSP deep tissue in vivo sound printing.
Exactly. It suggests we could be moving towards building tissues within the body, not just replacing them from the outside. It fundamentally changes things.
It really does. And for you, listening, maybe you're what we call the learner, someone who wants the core knowledge fast, those aha moments, but without getting totally overwhelmed. Well, this deep dive is for you. Because even if you don't have access to a wet lab, the computational side of this field, bioengineering, it's surprisingly accessible.
That's a key point. So much of cutting-edge science now relies heavily on computers, on modeling and simulation.
So our mission today is basically twofold. First, understand the science behind this in-body printing DISP.
And
Second, shine a light on the digital tools, the software that lets anyone with some technical background, maybe in coding or engineering, start, tinkering with these bioengineering ideas from their own computer.
We've looked at sources covering both the DISP tech itself and that whole digital infrastructure that supports it.
So the goal is to bridge that gap, isn't it, between this amazing futuristic idea and something you can actually explore computationally?
Let's try and make that connection.
Okay, part one then, DISP. Surgical-free tissue printing. It feels like a massive shift for regenerative medicine, creating tissues inside a living organism, bypassing the scalpel.
It really does. And the core innovation here, it seems to be this combination of special materials they call sono-inks and using ultrasound to activate them.
Sono-inks. Tell me more. They sound pretty high-tech.
They are. Think of them as injectable liquids. They flow easily at body temperature.
Okay.
But, and this is the clever part, when you focus ultrasound waves on them, they solidify. They actually turn into a solid structure, precisely where the sound hits.
Wow, so they're like smart materials responding to sound.
Exactly. A kind of smart material, yeah. It's designed to react specifically to that acoustic trigger.
So how does this get around the problems earlier ultrasound printing attempts had? I remember reading they were kind of fiddly, maybe too sensitive.
That's a really important point. Yeah, previous methods had limitations. The bioinks were often mechanically sensitive, meaning they could be easily damaged or wouldn't print accurately. Yeah. Plus, you had issues with slow printing speeds. The resolution wasn't always great. And there were concerns about the ultrasound generating too much heat, potentially damaging nearby tissues.
And biocompatibility, I suppose, making sure the body doesn't reject the ink.
Biocompatibility, too, absolutely. So these sono-inks were developed specifically to tackle those things. The stress sensitivity, the potential thermal damage, biocompatibility. It's a neat example of materials science meeting acoustic engineering.
Like piezoelectric materials responding to pressure or shape memory alloys, that kind of tailored response.
Sort of, yeah. It's that idea of engineering a material to have a very specific controlled response to an external stimulus, in this case, ultrasound. Overcoming those earlier hurdles was key.
Okay, walk me through the process then, step by step. How does it work? Inject liquid, get tissue.
It's pretty elegant, actually. First, you formulate the bioink. It's got molecule chains programmed to link up, tiny fatty microbubbles containing binder molecules.
Binders.
Yeah, they're the trigger for solidification. Oh. And often they add chemicals that help visualize the process, scatter sound, or light up. This ink gets injected, maybe directly or using a thin tube, a catheter, to reach deeper spots.
Okay, ink's in place. Then what?
Then step 2, focused ultrasound. You aim the sound waves right at the ink. The sound energy ruptures those fatty bubbles.
Up.
Yeah, basically. That releases the binders. The binders then initiate a reaction, causing those molecule chains to interconnect, link up, and solidify. That's the printing action.
You can watch this happen.
That's step 3, real-time monitoring. There's another molecule in the ink that's sensitive to ultrasound, allowing them to track the tissue formation. Plus, those visualization chemicals we mentioned scatter sound or illuminate, giving another view.
Crucial for safety and accuracy, I'd imagine.
Absolutely vital. And finally, step 4, cleanup. Any unsolidified ink is designed to be biocompatible. The body can metabolize it, eliminate it, or sometimes a treatment similar to one used for heavy metal poisoning can dissolve it.
Wow. So the ink isn't just a building block. It solidifies, it monitors itself, and cleans up after itself. That's a lot for an ink to do.
It is. It's a multifunctional composite material. Controlled solidification, monitoring, clearance, all built in. It reminds me a bit of how integrated circuits revolutionized electronics by packing multiple functions onto one chip.
And that real-time monitoring, you mentioned it's a foundation for the future.
Yeah, exactly. While the current system might rely on the operator watching, having that real-time feedback inside the body is a huge step. It paves the way for future automated systems, maybe closed-loop feedback control where the machine adjusts itself based on what it sees.
So self-correcting bioprinting.
Potentially, down the line. That seems to be the goal.
And they're not just printing, like, simple blobs, are they? Their article mentions some advanced applications in animal models.
Oh, this is where it gets really exciting.
Yeah.
They've demonstrated more than just basic tissue shapes. In rabbits and mice, for instance, they've printed soft biosensors inside the body.
Biosensors, how?
By including conductive nanoparticles in the sono ink. Imagine having sensors inside you for continuous monitoring diagnostics.
Wow.
And they've also created medication depots, basically printing little reservoirs loaded with drugs, anti-cancer drugs, and antibacterial ones right where they're needed.
And you trigger the release with ultrasound, too.
Exactly. Targeted, controlled drug delivery. Plus, they managed to restructure tissues several inches deep in rabbits, which suggests this isn't just a surface technique. It has potential for scalability.
So DISP isn't just one thing, it's more like a platform.
Precisely. A platform technology. The core is the ultrasound-triggered solidification. But by changing the payload in the Sono ink, different cells, nanoparticles, drugs, whatever, you can adapt it for many different applications.
Like a semiconductor fab making different chips using the same basic process.
Good analogy. Yeah, very much like that.
Okay, so we have this incredible internal printer. But how do scientists actually design these complex structures and figure out the printing parameters? That's where the computers really come in, right? Part 2, the digital scaffolding.
Absolutely. DISP and other advanced biofabrication techniques lean heavily on a whole ecosystem of computational tools. And this, I think, is where it gets really accessible for listeners with, say, engineering or computer science backgrounds.
Because the skills are transferable?
Largely, yes. It provides a really interesting entry point without needing the physical lab setup. Initially, anyway.
So it starts with design, right? Like CAD for regular 3D printing, designing life.
Exactly. Computer-aided design, CAD, is fundamental. Just like architects design buildings, bioengineers use CAD to define the architecture of tissues, scaffolds, the structures they want to print.
Are there specialized tools for this?
Oh, yes. Many bioprinter companies offer their own software. Reagan has Shaper, for instance. It lets you design complex 2D, 3D structures, define material properties, even manage projects.
Okay.
Celllink's DNA Studio has a shape generator tool. It simplifies creating common biological shapes like lattices or cylinders. Makes it easier if you're not a CAD guru but need those basic building blocks.
That makes sense. What about making something specific for a patient, like using their own scan data?
That's a huge area, personalization. Software like TSIM from Advanced Solutions can actually import CT or MRI scan data
Directly from the hospital scan.
Pretty much. It lets you design a tissue construct or a device that's tailored perfectly to that individual's anatomy. It's a big step towards personalized medicine. Think reverse engineering, but for biology.
And you mentioned simplifying tools like that shape generator. Is that a trend, making it easier for biologists, maybe?
Definitely. Higher levels of abstraction in BioCAD. It lets researchers focus more on the biological function they want to achieve rather than wrestling with complex geometry software.
Like using libraries and programming. You don't need to know how the square root function works, just that it does.
Exactly like that. Companies like Aspect Biosystems even talk about computational tissue design being central to their whole platform. It really underscores how critical the digital design phase is.
Okay, design done. But printing delicate bioinks with living cells, how do you know it'll work and that the cells will survive? Simulation time. Simulating the unseen.
Absolutely. Simulation is crucial. It lets you predict and optimize in silico on the computer before you waste precious time and, frankly, expensive biological materials in the lab.
Saves resources. What kind of simulations are we talking about?
Well, first, you need to translate the CAD design into instructions the printer understands. That's slicing and G-code.
Like standard 3D printing.
Just like it. The model gets sliced into layers, and G-code tells the printer exactly where to move, when to deposit ink, etc. Software, like Celllink's DNA Studio 4, often includes G-code editors and preview modes.
So you can watch a virtual print happen.
Exactly. You see the printhead path, where the material goes. It helps catch potential errors, collisions, gaps, inefficient movements before you even load the bioink. There are standalone G-code simulators, too, like Mesh Inspector Online. It's like proofreading the instructions.
Smart. But the ink itself, it's not just plastic, right? It's complex, fluid dynamics.
Right. Bioinks are often non-Newtonian fluids. meaning their viscosity changes under stress. Plus, they contain living cells. Understanding how these inks flow through tiny micronozzles is critical for cell viability and print accuracy.
Hard to see that experimentally, I bet.
Very hard at that scale. So computational fluid dynamics, CFD, becomes invaluable. CFD simulations give you insights into shear stress on the cells. Too much stress can kill them. Pressure gradients, velocity profiles.
So you can optimize the nozzle shape, the ink recipe, the print speed.
All of the above. Optimize nozzle geometry, bioink rheology, its flow properties, printing parameters like pressure and speed, all to minimize cell damage and ensure the final structure is accurate. CFD acts like a virtual microscope for fluid flow.
Okay, CFD for flow, and for DISP with the ultrasound, simulating sound waves too. The power of sound simulation.
Logically, yes. For DISP, simulating the acoustic field is crucial. You need to know exactly where the ultrasound is focusing and at what intensity to trigger solidification without harming surrounding tissue.
Is that a mature field, simulating medical ultrasound?
Oh, yes. There's a lot of work in simulating ultrasound for imaging, diagnostics, therapy planning, even training simulators for doctors. Tools like SautoSim, CAE Vimedix, Bodyworks Eve, Display US exist. Some are software-only, cloud-based.
Any open source options?
There are. Things like the PLES toolkit for ultrasound-guided interventions or Focus for calculating transducer pressure fields. While maybe not plug-and-play for DASP specifically, the underlying physics simulation capability is there. You'd need sophisticated models for DISP to get that acoustic focus just right.
It sounds like you often need to simulate multiple things interacting at once, right? Fluids, sound, heat, material changes.
You've nailed it. That's the need for multi-physics simulations. Processes like DISP inherently involve coupled physics, fluid dynamics, acoustics, material phase transition, liquid to solid, biological responses.
So you need tools that can handle all that interaction.
Increasingly, yes. To really capture the complexity, you need multi-physics simulations. There are general-purpose open-source frameworks like 4C or SV Multiphysics used for complex engineering problems. They're already being applied to things like additive manufacturing, analyzing thermal stress, or particle distribution in metal 3D printing, for example. The principles are transferable to bioprinting's complexities.
Okay, this is fascinating, but maybe still feels a bit abstract for someone listening thinking, what can I do with my computer? How do we bridge that gap? Part 3, your at-home computational biolab.
Right. This is the really empowering part. You don't necessarily need a fume hood and pipettes to start exploring bioengineering concepts, especially if you have a background in engineering or computer science.
It's about recognizing familiar concepts.
Exactly. Foundational concepts. Think about how you represent things digitally. In CS or engineering, you use meshes, voxels, particles. Right?
Sure, for 3D models, simulations, games.
Well, bioengineering uses the same ideas. Meshes define organ surfaces, tissue scaffolds, even large biomolecules. You can use software like 3D Slicer to generate meshes from medical scans. Voxels' 3D pixels are fundamental to interpreting those scans, understanding internal structure.
Okay.
And particles are agents. We use those to simulate dynamic systems, cells, moving, interacting. Agent-based modeling, ABM, is built on this. Think of Tissue Forge. These are representations you might already be comfortable with.
So the representation is similar. What about simulating behavior? Growth, interaction.
Again, familiar paradigms apply. Cellular automata, simple grid models with rules, can show basic growth patterns. Agent-based modeling, as I mentioned, treats cells as autonomous agents with rules for division, movement, death, interacting with their environment.
Like simulating a little virtual tissue.
Precisely. Then there are reaction diffusion systems, which model how chemical concentrations change over space and time. Think pattern formation, cell signaling. Virtual cell, V-cell, is a tool for that. And don't forget, biomechanics cells respond to forces. CFD can simulate shear stress. Particle models in Tissue Forge can simulate mechanical interactions.
So if you know simulation techniques from other fields, the leap isn't necessarily huge?
Exactly. It's often more about learning the biological context and the specific software interfaces rather than entirely new computational principles.
That's really encouraging. Let's talk tools then. What's in the open source arsenal for the aspiring biotinkerer?
There's a fantastic amount of powerful, free, open source software out there. Your computational biolab starter kit, if you will. Okay, hit me. We mentioned 3D Slicer, great for medical image visualization, segmentation, isolating organs, turning scans into 3D models. It even has AI tools now. First project idea, grab a sample CT scan online and try segmenting an organ.
Cool. What else?
Blender. It's a general 3D powerhouse, but with the molecular nodes add-on, you can visualize complex molecular structures like proteins, Download it from databases like the PDB. First project. Download a protein file and just explore its 3D shape in Blender.
Nice. For simulating tissues?
Tissue Forge. It's particle-based, good for modeling cell aggregation, spheroid fusion, nutrient diffusion. It integrates with Python and Jupyter notebooks, which is great for scripting. First project. Simulate 2 simple cell clusters merging.
What about the fluid dynamics, the bio-ink flow?
OpenFoam. It's a professional-grade CFD toolbox. Steep learning curve, maybe, but very powerful. The U.S. National Institute of Standards and Technology, NIST, actually has some benchmark data for bio-ink flow you could use. First project. Model simple fluid flow in a microchannel.
Okay. Any others?
4C for multiphysics, if you want to tackle complex couple problems later on. VCell for modeling cellular biological systems, signaling pathways, reaction networks. SymBioPython and Synergetica for synthetic biology designing genetic circuits. And ZellDetective, a Python tool for analyzing time-lapse microscopy images, tracking cells.
Wow, quite the toolkit. We should probably summarize these somewhere for people.
Definitely. We have a table listing their uses, and then maybe a simple first experiment for each.
Perfect. So those project ideas you mentioned, let's recap a few to give people concrete starting points.
Sure. 5 ideas. 1, design a virtual tissue scaffold. Use 3D Slicer to get a basic shape, maybe FreeCAD or Blender to design the porous structure. You learn CAD for bio scaffold design principles. Simulate bio-ink flow. Use OpenFoam and maybe that NIST dataset to model flow in a micronozzle. Learn basic CFD. Understand shear stress implications for cells. 3, visualize protein interactions. Use Blender with molecular nodes. Download protein structures, PDB files, relevant to bioinks. See how they might fit together. Learn molecular visualization, protein structure basics.
4,
simulate cell aggregation. Use Tissue Forge. Model how individual cells following simple rules can clump together or form layers. Learn agent-based modeling, basic tissue self-organization principles.
and 5.
Basic genetic circuit simulation. Use Synergetica or another tool like GrandS. Design a simple logic gate like an AND gate using simulated genes and proteins. Learn synthetic biology concepts, genetic circuit design.
These sound like great ways to learn by doing. Again, we can put these in a table. What about coding? You mentioned Python?
Python is huge in this space. Bioinformatics, computational biology, bioengineering, its readability, versatility, and amazing libraries make it dominant.
What are the essential libraries?
Your core toolkit would be NumPy for numerical operations, Pandas for data handling, Matplotlib and Seaborn for plotting, and SciPy for scientific computing. Most engineers or data scientists will know these.
And biospecific ones.
BioPython is the classic. PySam for working with sequencing data, Scampi for single-cell analysis, BioTite for structural bioinformatics. There are tools like TIMI for ecological evolutionary modeling, GrandS for simulating gene regulatory networks.
What about AI and machine learning?
Definitely relevant for more advanced work. Scikit-learn for general machine learning, TensorFlow and PyTorch for deep learning. Analyzing complex biological data often benefits hugely from ML.
So familiarity with Python is a massive advantage.
Absolutely. It lowers the barrier to entry significantly.
Where can people go to learn more or connect with others? Courses, communities?
Loads of options. Online platforms like Coursera, ADX, Class Central have courses. Look for titles mixing biology and programming, like Biology Meets Programming or Python for Genomic Data Science.
And communities?
The open source communities are key. The 3D Slicer Forum, Tissue Forge GitHub page, OpenFoam community. They have forums, documentation, tutorials, active users willing to help. Broader communities like Hackteria, DIY Biospaces, Fab Labs often have bio-interests. And platforms like Galaxy Project provide accessible bioinformatics workflows.
Specialized platforms, too.
Yeah, things like Saturn Cloud, Terra, DNNX's, 7 Bridges, Latch Bio. These often offer cloud environments with tools pre-installed, sometimes focused on specific data types like genomics. Plus, databases like the UMD Bioink database are crucial resources. The collaborative spirit is strong.
Okay, let's zoom out a bit. Part 4, the bigger picture, all this computational access.
Yeah.
It feels connected to broader trends, doesn't it? Open science, citizen science.
Very much so. The democratizing force of open source.
All right.
Open source software, like the tools we've discussed, dramatically lowers the barrier to entry. Think about open source hardware, too, like low-cost bioprinters designs the PrintS project is one example.
Yeah.
Resources like the UMD Bioink database, which catalogs properties of different bioinks, or open simulation frameworks like 4C, they foster collaboration. People can build on each other's work much more easily. It really enables participation from diverse backgrounds, not just established labs.
And this connects to the whole citizen scientist idea, DIY biology, computational biohacking.
Absolutely. The rise of the citizen scientist, the DIY bio movement shares a lot with hacker culture, biohacking, wetware hacking. The motivation is often about reducing costs, education, making science more accessible outside traditional institutions.
We see that with open source lab equipment designs too, right? 3D printed centrifuges, PCR machines.
Exactly. Those enable the wet lab side. But increasingly, computational biohacking is central. Exploring in silico lowers the barrier even further than setting up a home lab. You just need a computer and the right software.
It's powerful. But with power comes responsibility, right? Even in this simulated world, there must be ethical considerations, navigating the frontier.
Crucial point, yes. Even if you're only working with code and data, ethics matter. Data privacy and security is a big one, especially if you ever handle patient-derived data, even anonymized. Encryption, secure storage, understanding regulations like HIPAA or GDPR are important.
What else?
Algorithm bias. If the data used to train models is biased, the simulations or predictions can be inaccurate or unfair, potentially leading to health disparities down the line. Transparency in models and using diverse data sets is key.
And the potential for misuse, responsible innovation.
That's the dual-use concern. Could models or designs be used for harmful purposes? The DIY bio community often emphasizes transparency to mitigate this, but it's a concern even for computational work. Think about simulating pathogens or toxins.
We also need to ensure the work remains focused on benefiting people, right?
Human-centeredness, yes. Respecting human life and dignity in the designs and goals of the work. And there's the tension between intellectual property and openness.
Yeah.
How do you balance protecting innovations with sharing knowledge to accelerate progress? Licensing, proper attribution are part of this.
And finally, remembering that simulations aren't reality.
Exactly. Accuracy and interpretation. Models are simplifications. You have to understand the assumptions, the limitations, avoid overstating conclusions based on simulations. There was that case with AI potentially making it easier to design harmful molecules. It highlights the need for critical thinking about simulation outputs.
So ethical mindfulness, responsible conduct, critical thinking, they're essential parts of the toolkit too.
Absolutely. As you explore, keep these considerations in mind.
Okay, wrapping up then. This journey through in-body bioprinting, like DISP, it's genuinely exciting technology.
The potential is enormous, yeah.
But the big takeaway for you, our listener, might be that the computational side of bioengineering is wide open for exploration. Your existing technical skills are likely very transferable.
That's right. Tools like Blender for visualization, Tissue Forge for simulation, 3D Slicer for working with medical data, they're accessible. Project ideas like designing a virtual scaffold or simulating cell behavior are totally doable on a personal computer.
So the encouragement is dive in, embrace project-based learning, leverage those Python libraries we talked about, and definitely connect with the online communities.
Don't be afraid to start small and build from there.
And maybe a final thought to leave you with. As you start this computational biotinkering, consider the potential, not just to learn, but maybe even to contribute in some small way to understanding life, to creating new bio-inspired solutions. The power to model and simulate biology computationally is incredible.
But always wield that power responsibly. Keep those ethical considerations front and center as you explore this amazing frontier.